CRISPR-Cas9 technology. It was in news few years before, and UPSC has asked question on the CRISPR-Cas9 technology also as well. CRISPR-Cas9 technology has awarded for Nobel Prize in Chemistry. So this is very important research in the medicine field. So for curing the cancer, this gene therapy model will be used. So here we will discuss about the how gene therapy could cure cancer. Who lead the trial for this form of experimental medicine? How does base editing help in curing cancerous T cell? How does the CRISPR-Cas9 system work? What was the treatment a complete success? Does base editing treat blood disorder as well? So this is the some question, and here we will try to find out the answer of this question. Scientists in the United Kingdom testing a new form of cancer therapy reported success in a teenage girl, Alicia, with a form of cancer cell T cell acute lymphoblastic leukemia. What happened in the form of cancer? In this form of blood cancer, the T cell, which are a class of white blood cell equipped to hunt and neutralize threat to the body, turn against the body and end up destroying healthy cells that normally help with immunity. The disease is rapid and progressive and is usually treated by chemotherapy and radiation therapy. What marked Alicia treatment? The BBC reported that Alicia, 13-year-old, tried several of a standard treatment including chemotherapy and radiation but limited success. Just when it seemed there was no hope, she was enrolled in a trial testing of an experimental medicine. This trial was led by doctors and scientists at the University College of London and Great Ormond Street Hospital. Alicia was the first to receive experimental gene therapy that relied on a new technique called base editing. Here we will see that what is the base editing technology. A person's genetic code is several permutation of 4 bases. First, adenine A, guanine G, cytosine C, and thymine T. Sequences of these bases akin to letter in the alphabet spell out genes that are instructions to produce the wide array of protein necessary for the body's function. In Alicia case, T cell, perhaps because of misarrangement in the sequence of bases, had become cancerous. A way to correct this misarrangement could mean a healthier immune system. In the last 2 decades, the world of biomedical engineering has been enthused by a technique that allowed genes to be altered and errors fixed. This most popular among these approaches has been the CRISPR-Cas9 system. Inspired by how certain bacteria defend themselves against virus by snipping out and storing pieces of their gene, the CRISPR-Cas9 system consists of an enzyme that acts like molecular scissors. It can be made to cut a piece of DNA at a precise location, and a guide RNA can be used to insert a changed genetic code at the sites of incision. While there are a few ways to effect such changes, the CRISPR-Cas9 system is believed to be the fastest, most versatile system to effect such gene editing. David Liu of the Broad Institute of Broad Institute, Massachusetts has improved the CRISPR-Cas9 system to be able to directly change certain bases. Thus, a C can be changed in the G and T into an A. While still a nascent cell, nascent technology-based editing is reportedly more effective at treating blood disorders which are caused by so-called single-point mutation or when a change in a single base pair can cause terminal disease. How did base editing work for Alethia therapy? The objective of the gene therapy in the case of T-cell leukemia was to fix her immune system in a way that it stops making cancerous T-cell. First, healthy T-cells were extracted from a donor and put through a series of edits. The first base edit blocked the T-cell targeting mechanism, so it would cease attacking Alethia's body. The second removed a chemical marking called CD7, which is an alt T cell, and third, prevented the cells being killed by a chemotherapy drug. Finally, the T cells were programmed to destroy all cells, cancerous or protective, with CD7 marked on it. After spending a month in remission, she was given a second donor transplant to grow her immune system that would contain healthy T cell. How effective was the treatment? 3 months after the treatment, her cancer seemed to resurface, but the most recent investigations suggest no sign of it. According to BBC, Alethia was one of the 10 people enrolled in the trial to receive the treatment. It has been 1.5 years since she was first diagnosed with the disease, and whether the treatment has reliably and entirely fixed her immune system remains to be established. So this was the CRISPR-Cas9 technology and about the cure of the cancer. So stay connected with us. So subscribe our channel Satyavan Anand. Thank you.